Hi, my name's Cole, and today we're going to be talking about DNA and protein synthesis. So before we can understand protein synthesis, we must understand what DNA is. So DNA is made up of a sugar, a base, and a phosphate. Those three things put together, it creates a nucleotide. And DNA is just many of these nucleotides bonded together. With all these nucleotides bonded together creating DNA, that's what creates all the genetic information for all the somatic cells. Then there's RNA. There are three different types of RNA. There's tRNA, which is made up of 3' amino acyl acceptor and an anticodon loop made up of 3 nucleotides. Then there's rRNA, which is made up of 2 pieces, a 30S ribosomal subunit and a 50S ribosomal subunit. Lastly, there's mRNA, which is the genetic template made from the DNA. The first step in protein synthesis is transcription. This is where a section of the DNA is unwound, revealing the nucleotides. Then the enzyme called helicase will come in and attach itself to the DNA, spinning it to unzip the DNA. Once the DNA is unzipped, a piece of mRNA will come in, matching up with the nucleotides on the DNA with the corresponding mRNA nucleotides. Once this is done, the mRNA will go into the cytoplasm. It enters the cytoplasm through a nuclear pore and finds its way to a 30S ribosomal subunit. Once it has found a 30S ribosomal subunit, it will bind to it, which this will become the workbench of the protein synthesis. Once it is bound, this triggers a tRNA with the matching start codon to the mRNA to bind to it. Once the tRNA has bound to it, this triggers another reaction for a 50S ribosomal subunit to bind to the 30S ribosomal subunit. Once this has happened, the 2 ribosomal subunits bound together creates a functioning ribosome. Once the ribosome has been created, it can now move on to the process of translation. Translation is when tRNA bind to the mRNA, reading the nucleotides and the codons, and create an amino acid chain. There are 3 different sites during the process of translation. There's the P site, the A site, and the E site. But sometimes the E site and the P site are referred to as a hybrid site. At the A site, this is where new tRNA will come into the ribosome before moving to the P site and to bond with the amino acids. The P site is where the amino acids are bonded, and then it leaves one tRNA empty and with another tRNA with both of the amino acids. The E site is where old or used tRNA are removed from the ribosome to go find another amino acid. The process of translation is repeated until a stop codon is read, and then the process of termination is began. For termination, the amino acid chain is completely built, and the last codon on the mRNA reads stop. At this point, both pieces of tRNA, both pieces of rRNA, and the amino acid are all released from each other, leaving a newly created protein. This entire process doesn't always go so smoothly, and mutations happen. There are a couple different types of mutations, one of them being substitution. Substitution is when one of the bases gets switched out for another one, like A for G. Another one is insertion, when bases are just inserted into the chain, such as TTG into our chain. Another type is deletion, where it's the opposite of insertion and ones are taken out, such as GA. The last type is called the frame shift, where the codons are removed, leaving it incorrect in to be read in the wrong direction, such as the fat cat sat turning into gibberish. All of these are false protein codes. Well, I hope you enjoyed my explanation of protein synthesis. Remember to like, share, and subscribe, and I will see you guys next time. Happy reading week!